Ghost species, human relatives we only know from DNA, the silent revolution in paleoanthropology. For the vast majority of the history of science, the study of human evolution was a discipline of fragments. It was a science built upon the physical remains of the dead. Paleoanthropologists spent decades sifting through the sediment of caves and the dust of rift valleys, searching for calcified bone and mineralized teeth. The narrative of how we came to be was constructed exclusively from what we could see, touch, and measure with calipers. If a species did not leave a skull, a femur, or at least a distinctively shaped stone tool, it effectively did not exist. We built our family tree based on the hard evidence of fossils. We knew of Homo erectus, the Neanderthals, and the Australopithecines because they left their skeletons behind. However, in the 21st century, a profound shift occurred. The center of gravity in the study of human origins moved from the dusty trench of the excavation site to the sterile, clean room of the genetics laboratory. We entered the era of paleogenomics. With this technological leap, we began to see the invisible. We discovered that the human family tree is not merely a branching shrub of distinct physical lineages, but a tangled web of connections, many of which involve actors who have never been seen. These are the ghost species. They are populations of ancient humans that we have never found in the fossil record. We have no skulls to reconstruct their faces and no skeletons to estimate their height. We do not know what they look like or exactly where they lived. Yet we know they existed with absolute certainty. We know this because they left their shadows inside of us. Their genetic signatures persist in the DNA of modern humans, enduring through tens of thousands of years of inheritance. This text explores the scientific reality of these ghost species, the methods used to detect them, and the profound implications they hold for our understanding of what it means to be human. The Denisovan revelation. The story of ghost species begins properly in a cave in the Altai Mountains of Siberia. The site, known as Denisova Cave, had long been known to archaeologists. It contains a rich accumulation of sediment, preserving a record of occupation stretching back hundreds of thousands of years. In 2008, Russian archaeologists working at the site recovered a tiny fragment of bone. It was the distal phalanx of a pinky finger, so small it could easily have been lost in the sieve. Physically, the bone was unremarkable. It came from a juvenile, and based on the stratigraphy, it was roughly 40,000 years old. At the time, the assumption was that this bone belonged to a Neanderthal, or perhaps an early modern human, as both groups were known to have inhabited the region. The bone was sent to the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, for genetic analysis. The team there, led by Svante Pabo and Johannes Krause, had been pioneering the techniques required to extract fragile, degraded DNA from ancient specimens. When they sequenced the mitochondrial DNA from this finger bone, the results were shocking. The genetic sequence did not match that of modern humans, nor did it match the Neanderthals. It branched off from the common ancestor of humans and Neanderthals almost 1 million years ago. It was a completely new lineage. This was the first time in history that a new group of humans had been identified solely by their DNA. They were named the Denisovans. Unlike the Neanderthals, for whom we have hundreds of fossils, the Denisovians were initially known only from that single finger bone and a massive molar tooth found later. Yet their genome was sequenced to high coverage, providing a level of detail comparable to a modern medical test. This high-quality genome allowed scientists to compare the Denisovans to modern human populations. The results revealed that Denisovans were not an extinct dead end that vanished without a trace. They had interbred with the ancestors of present-day people. We found that indigenous populations in Papua New Guinea, Australia, and parts of the Philippines carry significant amounts of Denisovan DNA, ranging up to 5% of their total genome. This was the proof of concept for the ghost species. Here is a group of humans who spanned a vast geographic range, from the cold steppes of Siberia to the tropical rainforests of Southeast Asia. Yet they were practically invisible in the fossil record. If the Denisovans could hide in plain sight, what else was hidden in our genome? The methodology of hunting ghosts. To understand how scientists identify these phantom lineages, one must understand the mathematics of inheritance and the degradation of ancient molecules. DNA is a historical document. Every time a child is born, their genome is a mosaic of their parents' genomes. As we look back through generations, the blocks of DNA inherited from ancestors get broken up by a process called recombination. However, specific mutations occur at a predictable rate over time. By comparing the genomes of different populations, geneticists can calculate how long ago those populations diverged from a common ancestor. When geneticists sequenced the Neanderthal genome in 2010, they developed statistical tools to detect gene flow, which is the scientific term for interbreeding. One of the primary tools is known as the D-statistic, or the ABA-BA test. This test looks for asymmetries in the sharing of derived alleles. If modern humans from Africa and modern humans from Europe trace back to a common ancestor equally, they should share the same number of mutations with a Neanderthal genome by chance. However, if Europeans share significantly more mutations with Neanderthals than Africans do, it implies that Neanderthals contributed DNA to the ancestors of Europeans after they split from Africans. This was the smoking gun for Neanderthal admixture. To find ghost species, scientists take this logic a step further. They look for segments of DNA in modern or ancient genomes that are highly divergent. If a modern human carries a stretch of DNA that looks nothing like the rest of the human population's DNA and does not match the Neanderthal or Denisovan genomes, it must come from somewhere else. It must come from a lineage that split off from the main human trunk deeply in the past and then reintroduced that DNA later. These are introgression deserts and haplotype blocks. By measuring the length of these foreign DNA blocks, scientists can even estimate when the interbreeding happened. Long blocks indicate recent interbreeding, while short, chopped-up blocks indicate ancient events. This mathematical excavation allows us to reconstruct the existence of populations for which we have zero physical evidence. The deep African ghosts. While the Denisovans are a semi-ghost species because we eventually found a few fragments of bone, there are other lineages for which we have absolutely nothing but the statistical signal in modern DNA. Some of the most compelling evidence for these pure ghosts comes from Africa. Africa is the cradle of humanity, the place where our species originated and where we spent the majority of our evolutionary history. Consequently, African populations harbor the highest levels of genetic diversity on the planet. However, the preservation of ancient DNA in Africa is incredibly difficult. The hot, humid environment degrades the delicate, organic molecules of DNA rapidly. While we have recovered DNA from remains in Siberia that are hundreds of thousands of years old, recovering DNA from African fossils older than a few thousand years is a massive technical challenge. Therefore, the hunt for archaic human relatives in Africa relies almost entirely on statistical analysis of modern genomes. Several studies analyzing the genomes of West African populations, such as the Yoruba and Mende people, have found clear signatures of archaic introgression. Approximately 2 to 19% of the ancestry of these populations appears to derive from a ghost population that diverged from the ancestors of modern humans before the split between humans and Neanderthals. This ghost species would have split from our lineage roughly 625,000 years ago to 1.1 million years ago. This timeline suggests that while modern humans were evolving, likely from a stock resembling Homo heidelbergensis or a related group, this other lineage was persisting in parallel within Africa. They were likely distinct enough to be considered a different species, yet similar enough to mate with the ancestors of modern West Africans. The interbreeding event is estimated to have occurred relatively recently, perhaps as late as 43,000 years ago. This challenges the simplistic out-of-Africa narrative, which viewed Africa as a monolithic source of modern humans. Instead, it suggests that the African continent itself was a complex mosaic of different human populations, some modern, some archaic, interacting and interbreeding over hundreds of thousands of years. We have no name for this ghost species. We have no idea what they looked like. They might be related to fossils we have found, but cannot extract DNA from, such as Homo naledi or late surviving Homo heidelbergensis. Or they might be something entirely new, the Neandersovans and the super-archaic. The complexity of the ghost lineages increases when we look at the interactions between the archaic groups themselves. We tend to think of human evolution as us versus them, Homo sapiens versus the archaics. But the archaics were interacting with each other long before modern humans left Africa. When scientists analyzed the high-quality genome of the Denisovan from the Altai cave, they found something peculiar. The Denisovan genome contained segments of DNA that were far older than the Denisovan lineage itself. Roughly 1% of the Denisovan genome appeared to come from a much more ancient hominin. This DNA was extremely divergent, having split from the primate line leading to humans, Neanderthals, and Denisovans over 1 million years ago. The most likely candidate for this super-archaic ghost species is Homo erectus. Homo erectus was the first human ancestor to spread across Eurasia, leaving Africa nearly 2 million years ago. They survived in parts of Asia for a very long time. It appears that when the ancestors of the Denisovans migrated into Asia, they encountered these ancient populations of Homo erectus and interbred with them. This means that the Denisovans were not a pure species, but were themselves hybrids. This creates a dizzying Russian nesting doll of ghosts. We have modern humans who carry Denisovan DNA. That Denisovan DNA, in turn, carries trace amounts of super-archaic DNA. Therefore, billions of people alive today, particularly in East Asia and Oceania, carry tiny fragments of DNA from a species that might be Homo erectus, filtered through the Denisovans. This signal of the super-archaic is one of the deepest genetic links we have to the distant past. It bridges the gap between the early Pleistocene hominins and the late Pleistocene humanity. Furthermore, the interaction between Neanderthals and Denisovans was not rare. In 2018, the field was stunned by the publication of the genome of Deni, a bone fragment formerly known as Denisova 11. This individual was a first-generation hybrid. Her mother was a Neanderthal, and her father was a Denisovan. Finding a first-generation hybrid in the sparse fossil record is statistically improbable, unless such matings were reasonably common. The existence of Deni proves that the boundaries between these species were porous. It forces us to reconsider the very definition of species in the context of human evolution. If ghosts and known lineages were constantly swapping genes, they were perhaps better understood as a single, highly diverse metapopulation rather than distinct biological entities. The ghosts of island Southeast Asia. Nowhere is the mystery of ghost species more prevalent than in island Southeast Asia. This region, comprising the archipelagos of Indonesia, the Philippines, and Papua New Guinea, is a biogeographical laboratory. During the ice ages, lower sea levels connected many of these islands into a landmass known as Sunda, while others remained isolated by deep ocean trenches, forming the area known as Wallachia. This isolation drove the evolution of unique fauna, such as dwarf elephants and Komodo dragons, and it did the same for humans. The fossil record here is sparse but intriguing. We have Homo floresiensis, the hobbits on the island of Flores, and Homo luzonensis in the Philippines. These were small-bodied archaic humans who survived until relatively recently, perhaps 50,000 years ago. However, we have not yet been able to extract DNA from these tropical fossils. The heat and humidity destroy the genetic material. Despite the lack of ancient DNA, the modern genomes of people in this region tell a story of multiple ghost populations. When geneticists analyze the Denisovan DNA found in Papuans and East Asians, they find that it is not a single uniform signal. The Denisovan DNA in a Papuan is genetically distinct from the Denisovan DNA found in the Altai fossil. In fact, recent analyses suggest that there were at least 3 distinct lineages of Denisovans, which are as different from each other as Neanderthals are from Denisovans. One of these lineages, sometimes called D2, is found primarily in Papuans and Australians. Another, D1, is found in Asia. Some researchers have proposed that one of these Denisovan lineages might actually correspond to the physical fossils of Homo luzonensis, or Homo floresiensis. If this hypothesis is correct, these island hominins are not truly extinct in the genetic sense. They are ghosts living on in the indigenous populations of the region. The complexity of the data suggests that when modern humans arrived in Southeast Asia around 50,000 years ago, they did not enter an empty land. They entered a world teeming with different kinds of humans. Neanderthals in the west, multiple Denisovan-like groups in the east, and perhaps other, smaller archaic groups on the islands. They mated with several of them. The genetic history of this region is a palimpsest of these encounters, a layer cake of introgressions that we are only just beginning to deconvolute. The functional legacy of the ghosts. Why does this matter? Is the study of ghost species merely a stamp-collecting exercise, a way to fill in the blank spots on a map? It is far more than that, because the DNA we inherited from these ghosts is not junk. It is functional. It shapes who we are today. When modern humans migrated out of Africa, they were entering environments they were not biologically adapted to. They moved into the freezing cold of Eurasia, exposed to new pathogens, new diets, and new climates. The archaic humans, the Neanderthals, Denisovans, and the ghosts had been living in these environments for hundreds of thousands of years. They had already evolved the necessary genetic adaptations. Interbreeding provided a shortcut for evolution. Instead of waiting for beneficial mutations to arise spontaneously, modern humans acquired them through admixture. This process is known as adaptive introgression. The most famous example involves the Tibetan people. Life at high altitude is difficult due to low oxygen levels. Most humans respond to high altitude by producing more red blood cells, which thickens the blood and can lead to cardiovascular problems. However, Tibetans have a genetic variant of the EPAS1 gene that allows them to thrive in thin air without thickening their blood. When scientists traced the origin of this gene, they found it matched the Denisovan genome perfectly. The ghosts of the past gave the people of the Himalayas the ability to live on the roof of the world. Similarly, ghost DNA has played a massive role in our immune systems. The HLA-distinct genes, which are critical for recognizing and fighting pathogens, show high levels of archaic introgression in modern Eurasians and Papuans. We inherited a chemical library of defenses against local viruses and bacteria from the ghosts we encountered. However, the legacy is not always positive. Some of the DNA we inherited is maladaptive in the modern world. Variants associated with risk for depression, blood clotting disorders, and allergies have been traced back to Neanderthal and Denisovan ancestors. In the context of the Pleistocene, quick blood clotting might have saved your life after a hunting injury. Today, it increases the risk of stroke. A hyperactive immune system might have saved you from prehistoric intestinal worms. Today, it causes hay fever. We are walking chimeras, carrying the biological strength and weaknesses of species that no longer exist. The problem of identifying the source. One of the significant challenges in the study of ghost species is the problem of attribution. When we see a strange segment of DNA in a modern genome, how do we know exactly who it came from if we don't have a reference genome? We can tell it is archaic, but giving it a name is difficult. This is where the field relies on complex modeling. Scientists simulate different scenarios of human evolution on supercomputers. They run millions of simulations with different population splits, migration rates, and interbreeding events to see which model best fits the actual data observed in modern DNA. This is how the super-archaic contribution to Denisovans was inferred. No other model could explain the pattern of derived alleles. However, these models have limitations. They assume that we know the basic architecture of the population tree. If there were multiple ghost species interbreeding with each other before mixing with us, the signal becomes incredibly muddied. We are also limited by the lamppost effect. We look for ghosts where we have the light of modern genomic data. Large parts of the world, including vast areas of Africa, the Middle East, and India, are undersampled in terms of high-quality whole genome sequencing of indigenous populations. As we sequence more diverse populations, we are likely to find more ghosts. There is a strong possibility that an unknown archaic human species contributed to the genomes of people in the Indian subcontinent or the Arabian Peninsula, but we simply haven't spotted the signal yet because we lack the reference data. The map of ghost species is currently drawn with large blank spaces that future research must fill. Sediment DNA, and the future of the ghost hunt. The future of finding ghost species may not rely on finding more bones. A revolutionary technique is changing the game: sediment DNA. Scientists have discovered that DNA can be preserved in the soil of cave floors even if no bones are present. Humans and animals shed DNA constantly, in hair, sweat, and waste. This microscopic detritus falls into the dirt and binds to mineral particles. By taking core samples of dirt from archaeological sites and sequencing the DNA found within, researchers can determine who lived in a cave and when. This method has already confirmed that Denisovans occupied the Baishia Karst cave in Tibet, a site where only a jawbone was found. It allows us to survey hundreds of caves rapidly. We can now look for ghost species in layers of earth where no fossils survived. This opens up the possibility of mapping the presence of these populations across time and space with a resolution previously unimaginable. Imagine a cave in West Africa where the soil reveals the genetic signature of the elusive African ghost species dated to 20,000 years ago. This would confirm their late survival without needing a fossil. The combination of sediment DNA and paleoproteomics, which studies ancient proteins preserved in teeth, offers a way to bypass the limitations of skeletal preservation. Proteins last longer than DNA. While DNA might degrade after 100,000 years in a warm climate, proteins can last millions. By analyzing the protein sequences in ancient tooth enamel, we can distinguish between different species. This technique was used to identify a Homo erectus specimen from Dmanisi, Georgia, and a Denisovan from a jawbone found in China. As these technologies mature, the ghosts will have fewer places to hide. The philosophical implications of being a hybrid. The discovery of ghost species forces a philosophical reckoning. For a long time, Western science and philosophy held a view of human exceptionalism. We were the wise man, Homo sapiens, distinct and separate from the brutes of the past. We viewed other hominins like Neanderthals as failures, evolutionary dead ends that were wiped out by our superior intellect and technology. The genetic reality destroys this narrative of purity. We are not a pure species. We are a mongrel species. We are the result of a braided stream of populations that separated and merged again. The ghosts are not just them, they are us. A part of the Denisovan who looked out over the Altai Mountains lives on in the cells of a woman in Manila. A fragment of the code that built the immune system of a West African ghost lives on in a man in Nigeria. The extinction of these species was not total. While their cultures, their languages, and their unique physical forms have vanished, their biological essence was absorbed into the survivor lineage. This suggests that the definition of human is far broader than we thought. It encompasses a variety of ways of being, a variety of cognitive styles and physical adaptations that were all compatible enough to produce fertile offspring. It blurs the line between extinction and assimilation. Did the Neanderthals really go extinct? Or did they just dissolve into the larger population of Homo sapiens? The answer appears to be a bit of both. The ghosts remind us that we are connected to the deep earth and deep time in a very literal way. We carry the history of the Pleistocene written in our blood, the mystery of the skull shapes. While DNA tells us about relationships, it is poor at telling us about appearance. We know the ghosts existed, but what did they look like? This is where the intersection of genetics and developmental biology becomes the next frontier. By looking at the methylation maps of ancient genomes, scientists can infer which genes were turned on or off. This field, known as epigenetics, has allowed researchers to make predictions about the anatomy of the Denisovans. In a groundbreaking study, scientists analyzed the methylation patterns of the Denisovan genome and predicted that they would have wide skulls, robust jaws, and a specific pelvic shape. When the Denisovan jawbone was later found in Tibet, its measurements matched the genetic predictions with uncanny accuracy. This offers a glimmer of hope for visualizing the pure ghosts. If we can isolate the specific haplotype blocks that come from the African ghost or the superarchaic, and if those blocks contain genes involved in skeletal development, we might be able to reconstruct their morphology. We might be able to predict that the African ghost had a heavy brow ridge or a unique dental pattern. We could potentially match these genetic predictions to orphan fossil skulls and bones found in the archaeological record that do not fit into any known species. There are many such fossils in China and Africa that are currently labeled as archaic Homo sapiens or Homo heidelbergensis, but might actually be the physical remains of the ghost lineages we see in the DNA. The ghost of the genome might eventually meet the ghost of the fossil record, resolving the mystery, the connection to language and cognition. One of the most tantalizing questions concerns the cognitive abilities of these ghost species. Did they speak? Did they have art? Since we have no archaeological context for the pure ghosts, no tools or campsites definitely linked to them, we must look to the genes they left behind. Some of the genes introgressed from archaic humans are involved in brain development. However, there is also evidence of negative selection in gene deserts around areas associated with language and complex cognition. For example, the FOXP2 gene, which is heavily involved in speech and language, shows very little evidence of archaic introgression in modern humans. This suggests that the version of the gene held by modern humans was so essential to our success that any archaic version was weeded out by natural selection. This pattern is found in many genes expressed in the brain. It implies that while we were compatible enough to mate with ghost species, there were significant differences in our neural architecture. The human operating system might be a distinct feature of our specific lineage, one that could not easily tolerate archaic code. Yet this is not a settled science. The Denisovan archaeological record includes sophisticated jewelry and tools, suggesting minds capable of symbolism and complex planning. If the ghosts were the intellectual equals of our ancestors, their absorption into our lineage is even more poignant. It represents a merger of minds, not just bodies. The complex web of the Middle Pleistocene. To truly grasp the scale of the ghost phenomenon, we must look at the era known as the Middle Pleistocene, roughly 700,000 to 100,000 years ago. This was the muddle in the middle, a time of chaotic evolutionary experimentation. During this period, the common ancestor of humans, Neanderthals and Denisovans, split up. The ghosts we detect today are likely the result of populations becoming isolated during glacial maxima, ice ages that created physical barriers of ice and desert, and then expanding and mixing during interglacials. The super-archaic signal suggests that even older lineages from the early Pleistocene were still wandering Eurasia during this time. Imagine a world where 4 or 5 distinct types of humans walked the earth simultaneously. In the forests of Europe, the Neanderthals hunted ambush style. In the expanses of Asia, the Denisovans and the remnants of Homo erectus roamed. In Africa, the ancestors of Homo sapiens lived alongside the archaic African ghosts. In the islands, the hobbits persisted. It was a Lord of the Rings world. Not in fantasy, but in biological fact. The ghost signals are the echoes of the interactions between these groups. They tell us that isolation was rarely permanent. Whenever climate allowed, these groups found each other. The drive to connect, to mix, and to mate seems to be a fundamental human trait, predating our own species. The ghosts are evidence that cosmopolitanism, the mixing of different peoples, is the rule of human history, not the exception. The limitations of the molecular clock. A technical caveat that haunts the study of ghost species is the molecular clock. This is the rate at which mutations accumulate, used to date the splits between populations. The clock is not a metronome. It varies. It can tick faster or slower depending on the generation time, how old parents are when they have children, and the overall mutation rate of the species. Uncertainties in the mutation rate can shift the estimated divergence dates of ghost species by hundreds of thousands of years. When we say the African ghost split 700,000 years ago, that is an estimate with wide error bars. It could be 500,000, it could be 900,000. Calibrating this clock requires ancient DNA from fossils with secure radiocarbon dates. As we find more anchor points, ancient genomes from known times, we can refine the clock. Currently, the ghostly divergence dates are our best estimates, but they are subject to revision. This fluidity is part of the excitement of the field. A single new genome can rewrite the timeline of human evolution. The dynamic nature of the genome. The study of ghost species also highlights the dynamic nature of the genome itself. The genome is not a static blueprint, it is a landscape of competition. Introgressed DNA from ghosts is not distributed randomly. It is concentrated in some areas and completely absent in others. As mentioned, the deserts of archaic DNA suggest that natural selection actively purged ghost DNA that was harmful to the hybrid offspring. This phenomenon, known as hybrid incompatibility, often affects the male reproductive system. The X chromosome in humans is notably poor in archaic DNA, suggesting that male hybrids might have had reduced fertility. This aligns with a biological rule known as Haldane's rule, which states that in the hybrids of diverging species, the sex with different chromosomes, in mammals, the male XY, is the most likely to be sterile or absent. The fact that we have any ghost DNA at all means that the female hybrids were likely the conduit. The ghost mothers raised hybrid children who grew up in human tribes, passing on their mitochondrial DNA, which we see in some ancient samples, and their nuclear DNA. Over thousands of generations, the male sterility issues were purged, leaving behind only the beneficial or neutral ghost DNA. This implies a gendered dimension to the history of our interactions with ghost species, a story of mothers and daughters bridging the divide between lineages. The ongoing search for the dragon. One of the most exciting recent developments involves the Dragon Man cranium, or Homo longi, found in Harbin, China. This massive skull represents a population that lived in East Asia at the same time as the Denisovans. Morphologically, it is distinct from Neanderthals and modern humans. Many paleoanthropologists suspect that Homo longi is the physical face of the Denisovans. If we could extract DNA from the Dragon Man's skull and it matched the Denisovan sequences, the ghost would finally have a face. However, until that DNA is recovered, Homo longi remains a fossil looking for a genome, and the Denisovans remain a genome looking for a fossil. There is a high probability that many of the ghost signals we see in Asian populations correlate to the Homo longi population structure. The integration of Chinese paleoanthropology into the global genomic synthesis is crucial here. For decades, Asian fossils were difficult to place in the family tree. Now, genetics offers a scaffold to unify these findings. The ghost species concept provides the glue that binds the Harbin skull, the Dali skull, and the Jinyushan specimen into a coherent picture of Asian human evolution, potentially revealing them as diverse representatives of the Denisovan/ghost lineage. The ethical dimension of paleogenomics. The hunt for ghost species also raises ethical questions regarding the study of indigenous DNA. The discovery of ghost signals relies heavily on the genomes of indigenous peoples in Australia, Papua New Guinea, Africa, and the Americas. Historically, these populations have been exploited by scientists who took samples without proper consent or community engagement. The narrative of ghost species must be handled with respect for the living descendants who carry that DNA. It is their heritage. Modern paleogenomics is increasingly moving towards a model of partnership, where indigenous communities are co-investigators. They are interested in their deep history, but they also want to ensure that their genetic information is not used to stigmatize them or challenge their land rights. For instance, the discovery that Aboriginal Australians have been in Australia for 50,000 years and carry unique ancient lineages reinforces their status as the first peoples. The science of ghost species can be a tool for empowerment, confirming oral histories of deep time and distinct origins. It validates the feeling of distinctiveness that many cultures maintain. However, scientists must be careful not to use terms like archaic in a way that implies inferiority. The ghost DNA is not primitive in a pejorative sense. It is a reservoir of ancient adaptation. The path forward. As we look to the future, the resolution of our genetic telescope will only improve. We are moving from sequencing genomes to understanding the proteome and the metabolome of ancient humans. We are beginning to apply artificial intelligence to scan genomes for patterns of introgression that are too subtle for standard statistical methods to catch. AI models can learn the complex signatures of deep introgression and spot fragments of DNA that came from a ghost species 200,000 years ago, even if that DNA has been broken down into tiny shards. This might reveal ghosts even older than the super-archaic. Perhaps we will find traces of a lineage that split off 2 million years ago, a genetic echo of Homo habilis or Homo rudolfensis, lingering in the code of modern humanity. The story of human evolution has changed from a simple line of progress to a complex network of interactions. We are the survivors, but we are also the vessel for the dead. The ghost species are not truly gone. They are woven into the double helix of every cell in our bodies. They shape our blood, our bones, and our ability to survive in a changing world. By studying them, we do not just learn about the past. We learn about the biological composite that is the modern human. The ghosts are with us, and the science of the 21st century is finally giving them a voice. The unexpected complexity of the Americas. While the focus of ghost species research has largely been on Africa and Eurasia, the Americas hold their own mysteries. The peopling of the Americas was once thought to be a simple migration of Siberians across the Bering Land Bridge. However, genomic analysis of Amazonian populations, such as the Surui and Caritiana, revealed a puzzling signal. These groups share a small but significant genetic connection with Australasians, populations from Papua New Guinea and Australia. This signal, dubbed Population Y, does not exist in other Native American groups or in the Siberians, who are the primary ancestors of Native Americans. How did DNA from the South Pacific end up in the deep Amazon? It suggests the existence of a ghost population in ancient East Asia that contributed ancestry to both the first Australians and a specific wave of migrants who crossed into the Americas. This ghost population has since disappeared from Asia, replaced by later waves of migration, but its legacy survives on opposite sides of the Pacific. This finding complicates the map of human migration significantly. It implies that the ancestral pool of Native Americans was more diverse than previously thought, and that there were structured populations in Beringia or East Asia that we have yet to identify archaeologically. The ghost of Population Y serves as a reminder that even in relatively recent human history, within the last 20,000 years, there are lost lineages that we can only see through the lens of genetics. The immune system as a museum of ghosts. To understand the daily impact of these ghost species, one must look closer at the major histocompatibility complex, MHC. This region of the genome is the most variable part of our DNA and controls how our immune system identifies foreign invaders. It is under intense evolutionary pressure. If a population lacks the genes to recognize a specific virus, it can be wiped out. When modern humans met Neanderthals and Denisovans, they were meeting species that had adapted to the local distinct pathogens of Eurasia for hundreds of millennia. The introgression of MHC variants was not just a minor event, it was a survival necessity. Studies show that the sheer diversity of HLA alleles in modern humans is partly due to this archaic admixture. We essentially outsourced our immune adaptation to the ghosts. For example, the HLA-B73 allele is common in West Asia but rare in Africa. It is likely a legacy of Denisovan admixture. Without these ghost genes, the colonization of the globe by Homo sapiens might have been halted by disease. We walk through the world protected by the shields forged by species that died out 40,000 years ago. This is a functional immortality. The ghosts are not just passive passengers. They are active participants in our biological defense, the mystery of the ghost Y chromosome. While we have found ample evidence of ghost DNA in the autosomes, non-sex chromosomes, and some in the mitochondrial DNA, the Y chromosome tells a different story. The Y chromosomes of Neanderthals and Denisovans have, until recently, been difficult to sequence. However, recent work suggests that the Y chromosomes of Neanderthals were actually replaced by a human-like Y chromosome relatively recently, around 370,000 to 100,000 years ago. But what about the ghosts? We have not yet found a ghost Y distinct from the human/Neanderthal lineage in modern populations. This absence is data in itself. It suggests that male hybrids between modern humans and the deep ghost lineages, like the African ghost or the super archaic, were likely infertile, or that their Y chromosomes offered no advantage and were lost to genetic drift. This reinforces the idea of quote-unquote purifying selection, acting against the male line. The male ghosts may be gone, but the female ghosts transmitted the autosomal heritage that remains. This creates a bias in our window into the past, where we see the history of hybridization largely through the lens of female-mediated gene flow and autosomal retention, masking the fate of the ghost men. The link to circadian rhythms. Another fascinating area of functional introgression involves our body clocks. Neanderthals and Denisovans lived at high latitudes in Eurasia, where days are long in summer and very short in winter. Modern humans evolved in Africa near the equator, where day length is relatively constant. When our ancestors moved north, their circadian rhythms were out of sync with the new environment. Genetic analysis has shown that variants of genes affecting the circadian rhythm, specifically those influencing being a morning person, were introgressed from Neanderthals. These variants likely helped our ancestors adjust their sleep-wake cycles to the seasonal variations of the north. So if you are a morning person who wakes up easily with the sun, you might be thanking a ghost species for that trait. It is a behavioral adaptation encoded in DNA, a subtle nudge from the past that dictates how we live our lives today. This connects the macroevolutionary scale of species extinction to the microscale of our daily habits. The cultural interaction hypothesis. While we cannot sequence culture, the genetic evidence of ghosts implies cultural interaction. You do not interbreed with a population you are constantly at war with, or at least, not to the extent that the genes become fixed in the population. The survival of hybrid children suggests a degree of social integration. It implies that these ghosts were viewed as people, potential mates, and members of the community. This challenges the killer ape hypothesis of human evolution. It suggests that the replacement of archaic humans was not a simple genocide. It was a slow process of absorption and demographic swamping. The ghost species were likely much smaller in population size than the incoming waves of Homo sapiens. Over time, they were simply outnumbered. Their genes were diluted, but their contribution remained. This paints a picture of the late Pleistocene as a world of complex social negotiations, where different types of humans shared landscapes, resources, and beds. The ghosts were neighbors before they were ancestors, the limits of detection, and the unseen ghosts. We must also consider the ghosts we cannot see. Our current statistical methods are good at finding ghosts that are sufficiently genetically distinct from us. If a ghost lineage was very closely related to us, diverging, say, only 50,000 years before the mixing event, it would be almost impossible to detect with current technology. Its DNA would look too similar to our own. Therefore, our current count of ghost species is a minimum estimate. There could be many more shallow ghosts, populations that were slightly distinct but have merged back into the mainstream so thoroughly that they are invisible. The family tree is likely even more reticulated than we realize. We are only seeing the branches that stuck out at sharp angles. The ones that grew parallel to us are hidden in the noise of natural variation. Conclusion. The study of ghost species has fundamentally transformed our understanding of human origins. We have moved from a linear model of evolution to a braided stream model. We now know that our genome is a palimpsest, written over by multiple hands. The Denisovans, the African ghosts, the super archaics, and the insular ghosts of Southeast Asia are not mere footnotes in history. They are ancestors. They provided us with the genetic tools to survive in new environments, from the high altitudes of Tibet to the pathogen-rich rainforests. They live on in our immune systems, our skin, our metabolism, and our skull shapes. The science of paleogenomics has allowed us to resurrect these lost relatives, not in body, but in data. As we refine our methods and sample more of the world's biodiversity, the number of ghosts is likely to grow. We are learning that to be human is to be a hybrid. We are the sum of a collective history that spans millions of years and multiple species. The ghosts are not haunting us, they are us. The final dot of this text marks not an end to the inquiry, but a pause in a story that is still being written by the nucleotides in our cells.